course, I usually talk about it after we talk about DNA regulation, because the book brings it up in two different places, here and there. And it makes a bit more sense later on in the course when you've talked about DNA regulation and have more of a chance to look at how the DNA works out. So this is not going to be on the exam. This is just a freebie associated with the fact that it works out nicely to time with this event. Okay? So I'll tell you when we get back into material that's examinable. Right? But what I want to do is I want to use this to talk about cancer, because cancer is very timely at this stage. Cancer is collectively a group of disorders that are associated with the loss of control of the cell cycle. All right? Cancer is not one entity. Cancer is basically a classification of a whole group of disorders that arise because the cell cycle, for one reason or another, has been lost. And if you think about how complex it would be to actually regulate all of that stuff, you can sort of see why you might actually have one false thing happen in the cell cycle can lead to a cell that starts growing out of control. Right? And if you look at cancer cells, one of the things you will find is they have a number of common characteristics. They could have arisen by for different reasons, which is why it's so hard to actually work with and treat cancers. But one of the main things is that they have a change in the plasma membrane and the cytoplasm. And a lot of this is that it starts to increase permeability. Now, when we've been talking about cells, one of the main things that we've been talking about is that the membrane and all these transmembrane proteins facilitate quite a bit of selectivity as to what is going back and forth across that membrane, right? They got a lot of control by virtue of having to go through gated pathways or things like this to get across the membrane. One characteristic is you start to see a breakdown of that in cancer cells. They start to have passage of molecules that they don't normally allow back and forth across the membrane. All right? The other is that they undergo abnormal growth and division. They are going through the cell cycle faster. Okay? And another component is that they start to have weakened cellular adhesion. Right? And cellular adhesion is usually a factor of the proteins that are binding between two adjacent cells, holding onto each other. This suggests that some of those proteins are not being made properly, and they don't fit that perfect lock and key mechanism anymore, and so they start to break apart. Okay? And the last component of this is that cancer cells, if they are not removed, can advance to the stage where they will become lethal to the organism where the cancer is occurring. All right? Now, what causes these? Genes that resort in abnormal cell cycles are what we call cancerous genes, which we call oncogenes. All right? Now, certain genes within the chromatin, within the DNA, have a greater potential of actually going out of control, or basically starting to facilitate abnormal cell cycles. And those genes that are kind of subject to, if you get a mutation in that gene, subject to going out of whack and starting to get this cell out of its normal cell cycle, are called proto-oncogenes. And these are genes that tend to regulate some aspect, normal regulation of the cell cycle, that if a mutation arises in them and they change, then it can convert that gene into what's called an oncogene. All right? Now, the key here is that the cells that are considered cancerous do not respond to the normal signals that are regulating the cell cycle. All right? So if you were looking at this, they might be amping up cyclin production in, say, the G1 phase, which starts to get them all active, and they start to have a massive amount of MPF activity too early in that G1S, G2 phase. All right? Now, another thing is, in many cases, that G1 checkpoint doesn't necessarily need to wait for some kind of external signal. Sometimes the cell will spontaneously and inappropriately kickstart that G1 checkpoint, pull the cell out of the G1 too early. All right? And so it's not responding to things like growth factors. It just grows without getting something that's saying you need to start growing. It just grows by itself. All right? Now, so the key here is that you're getting out of whack. You're getting out of phase. Now, if you have a normal tissue and it suddenly starts to essentially ignore the normal regulatory cycle, it has undergone something called transformation. Now, what the key for that transformation is could be a variety of sources, which is why it's hard to deal with cancers. But when that cell does go cancerous and it starts to get out of that cell cycle, it has a great potential to snowball, right? Because what's first going to happen is you're going to start with a tumor, and a tumor is just a mass of cells that are growing outside of the normal cell cycle. As long as that mass stays in a contiguous lump and it doesn't start invading neighboring tissues, we would call that a benign tumor. Once the cell starts to invade surrounding tissues, and especially when the cell starts actually breaking down and it's losing cells off of that mass, they're no longer holding tightly to that cell mass, that is when the cells or the cancer is said to have metastasized. And this is where it gets very dangerous because this is where that cancer can spread from one part of a body to another. Now, this is where the breast cancer example comes into it. If you have a tumor mass that appears in the breast tissue, then what can happen is as the single cell starts getting out of whack from its cell cycle, it will start to grow, and that tumor will typically be intact and will grow as a mass of small cells within that one tissue area. Right? But what's happening is it's going to be dividing at an abnormal rate compared to what it should be. And so it starts to build up a mass. Now, think of what's happening. If it's maybe, say, initiating its cell cycle before it has actually hit the G1 checkpoint, it starts to go through synthesis G2 prematurely. Right? If you have something that ramps it up and it hits that G2 checkpoint early, that's where it becomes really dangerous. It starts to divide, and it may not have gone through the entire process of the sort of proofreading, cross-checking that it's correctly copied the DNA sequence, right? The faster you whip through S2 and G2, the more likely you are to have mistakes when you're copying out your DNA. Mistakes tend to correspond to mutations, and it can then screw up the sequence in particular genes that you're reading, and that in turn will screw up things like the primary structure of the proteins you're making, and that can screw up the secondary folding, the tertiary folding, and now you don't have a functional protein, or you have a hyperactive protein that doesn't regulate itself properly. Instead of waiting for an allosteric regulator, it just does whatever the hell it wants without any kind of control. And as you do that, it might speed the system up a little bit more, and speed it up a little bit more. And so now you're starting to regulate, and you're starting to accumulate even more mutations. And then you're starting to affect not only the mutations on the proteins that affect the cell cycle, but also